So chapter 18 is microbial pathogenesis. We're going to cover a couple topics. So we're going to be looking at genetics of pathogenicity, evolution, and in particular, we're going to be looking at molecular tools that microbes use to not only cause the disease, but to also evade the immune system. So different strategies microbes can use within the host. So looking at attachment methods, whenever we look at pathogenesis, we have to see how microbes can infect their host. So attachment is really step one. How does a microbe come into contact with the host cell? That's what we're looking at here. So we have mechanisms for adhesion, for adhering to the host cell, or attachment, as we see here. We're going to be covering pili, different forms of pili on the next slide, but there's also fimbriae, fimbriae and pili, we can consider the same thing. We're also going to see some protein interactions or non-pilus adhesion as well. So here is a pilus. What we can see is it's made of identical subunits and it just extends. And the tip of it has an adhesion protein. So this adhesion protein can bind to host cell receptors, essentially. Looking at different types of pili, we have a type 1 pili versus a type 4 pili. So this appendage is solely used for attachment. We see it's these small hair-like structures extending from the microorganism. And over here, we can see the type 4 pili actually is used for movement, so a twitching motility. So it can pull and push itself on a slide. It can extend the pili and then pull the pili back so it can twitch around. When we talk about biofilm formation, when microbes come together and live in a communal scenario, they use pili in order to have this twitching motility to move around to find one another. Okay, so there's many different non-pilus adhesion mechanisms that cells use, but we're not going to go into detail with all of them here. So these are cell wall associated proteins. This is nothing to memorize, but a lot of these that we see here may be brought up later, but no need to memorize the list, just showing other mechanisms. So talking about biofilms, again, one of the initial steps of formation of a biofilm is adhesion to a solid surface and then twitching motility so cells can find one another. So biofilms are made up of over 500 different microorganisms living together. They attach to a surface. They secrete this extrapolymeric substance that holds them together and protects them from the outside environment. And it's a communal living scenario. This can protect them from things like antibiotics or any kind of anything else like disinfectants that we add. So it doesn't necessarily penetrate through a biofilm. So this is a protection mechanism against not only the host immune system, but any kind of detergents or any kind of chemicals that we use to kill microorganisms. Okay, looking at cellular pathogens. We have this slide and we have this next one for, extracellular pathogens. And this slide I think is a little bit more visual, so I like using this one, but this one outlines everything in a little bit more detail. So we're going to look at how extracellular pathogens can avoid the immune system. The first one we're looking at here is capsules. So here we see an encapsulated microorganism. When it's encapsulated like this, it prevents phagocytosis from occurring. So essentially, our own immune cells can't engulf that cell because it has a capsule around it. So that's one of the protection methods that we're looking at. We can also see varying surface antigens. So surface antigens are these small attachments on the outside of the cell. That's actually what our immune system recognizes as these small attachments on the outside of the cell. So if we vary the antigen structure, and this can be from genetic changes by mutation, our immune system may no longer recognize a pathogen. If we're originally infected with this pathogen with the red type of antigens, and all of a sudden it altered to this different blue type of structure, our immune system might not recognize it. So varying surface antigens is a way to avoid the immune system as well. And then we also have quorum sensing. So quorum sensing falls into quite a few things over here. Quorum sensing enables cells to communicate with one another. They wait till they're at critical mass. They wait till they're at a critical volume when they replicate. And then they're able to synthesize virulence factors together, again, toxic factors. So they wait until they're at a high enough number in order to launch an attack. And that's part of biofilm formation as well. We have quorum sensing as communication, essentially talking between cells that are gathering together. Here we see a couple other ways of evading the immune system, like secreting fake cytokines. Cytokines are an immune part of the immune signaling. And then we have microbial cells manipulating host cells to secrete cytokines as well. So manipulation of the host immune system. So these are just a couple of ways that we see extracellular pathways, essentially avoiding the immune system. We're going to go ahead and look at intracellular pathogens here. And again, we have another diagram to look at intracellular pathogens. But intracellular pathogens can really avoid the immune system because our immune system doesn't really operate within cells. It operates around cells. So if they're able to live within a cell, they're already avoiding a large portion of the immune system. So we have two different kinds of cells that can live in the immune system. We have facultative intracellular pathogens and obligate intracellular pathogens. Facultative ones, they can evade our host cells, but they can also survive outside the cell. So they can live inside the cell or outside the cell. They're facultative. Obligate, on the other hand, these need to live inside of a host cell. So we'll see that they have to stay within the cells. That is where they reproduce. That is where their metabolism is active, is inside the cells. So we're going to see a couple examples of intracellular pathogens here and the fates of the intracellular pathogens. So the general process that we'll follow is 1 through 3, and then we'll look at different fates of the cells. Here we have a microbial cell coming into contact with a host cell. We see it being engulfed, so that's a process of phagocytosis where the host cell takes in the microorganism. One fate that we can see here is that the host cell within its phagosome actually binds with the lysosome, and the lysosome typically would break down the bacterial cell. It would degrade the bacterial cell and kill the bacterial cell. But what we see here is that this cell is able to live in inclusion bodies, and it's able to continue living like this. Another example of these fates would be something like Salmonella on fate 2. Instead of having a lysosome merging with the phagosome itself, it prevents it from merging. So the phagosome remains on its own with the microorganism inside of it. And here we can see Salmonella moving outside of the cell and then being again engulfed into another cell as a phagosome. What can even happen following this, if we're moving through different cells and we're looking at cells of the immune system, Salmonella can actually move into cells like a macrophage that moves up into the lymphatic system, into the lymph nodes. And from the lymph nodes, it will be spread throughout our vascular system. So we can see it entering into a host cell, preventing a lysosome from merging with it. Typically, lysosomes would kill anything within the phagosome, so it prevents this from happening. And then it moves into other cells, which can move into the immune system and then get spread throughout the vascular system to all points in our body. So that was fate 1. It can live in inclusion bodies where this lysosome will not affect it. Fate 2, preventing the lysosome from merging. And then we see fate 3 over here. So in fate 3, we can see something like Shigella or Listeria. They actually break out of the phagosome and they build an actin tail. So it's this actin tail attached to it that is assembled, and it jets its way through cells. We can see it moving from one cell to the next cell with this tail. The next image we're going to see here is that actin tail. So here's Shigella, and this is that actin tail that's assembled, and this is how it's swimming through cells. It can escape the phagosome this way and push its way through the cells. Here we see survival within a phagosome. So that would be something like Salmonella, like we see here, living within the phagosome, reproducing within the phagosome, the cells itself. Okay, let's look at exotoxin modes of action. So looking at how we can affect the cell itself, we have the plasma membrane disruptions. So we have certain toxins that can break down the plasma membrane, certain toxins that can break up the cytoskeleton. Remember, that's the support of the cell. So if we break up the plasma membrane, we can either lyse open the cell and kill the cell, or we can just poke holes within the cell membrane as well for entry of microorganisms. Looking at things like protein synthesis disruption, if we disrupt protein synthesis, the cell will die if it can't produce proteins. What other ones do I want to cover here? It can alter signal transduction. So we have receptors on the outside of the cell. They can communicate all the way down to the nucleus and regulate DNA expression. So some toxins, some exotoxins can interfere with this process as well. I think those are a couple good ones to mention. So looking at exotoxins' mode of action, so let's look at an AB toxin. This is a pretty common toxin. It's made up of two units, the A subunit and the B subunit. As seen in this picture here, the B subunit surrounds the A subunit. We're going to see what actually happens here. The A subunit in the middle is toxic, and the B subunit is what enables it to bind to the host cell. So essentially, the A subunit is hidden by the B subunit. The B subunit allows attachment to the cell, binding to the cell. Then the A subunit enters and is a toxic factor that can affect the host cells. So it's kind of a 1-2 punch with the AB subunit. We also have alpha toxins as exotoxins. Alpha toxins actually make a 7-membered ring. And we can see this ring here. This moves through the membrane of the cell. So here's a cross section. And because it moves through the membrane of the cell, it creates a pore. And this can cause the cell to lyse. So it can break the membrane. So it opens up a hole in the membrane. And then we lose osmotic regulation. So the cell will essentially burst open. We can see that with alpha toxins here with beta hemolysis of Staphylococcus aureus. This is red blood cells. This is sheep red blood cells in the agar. And with Staphylococcus aureus, it's actually releasing this alpha toxin, which is breaking open these red blood cells. It's bursting the red blood cells open. Okay. Looking at other exotoxins modes of action, we can talk about signal disruption. So we mentioned that a little bit earlier. I won't go too much into detail, but again, there's many signaling methods that can be altered with exotoxins. So it can disrupt normal functioning of the cell, whether it's going to be protein production, lipid production, transcription, translation, anything like this can be disrupted by these exotoxins in cell signaling disruption. Let's go ahead and look at the anthrax toxin. So anthrax is maybe something you've heard of. Anthrax was a bioterrorist weapon during 9/11. I know many of you were probably not born then, but for people like myself who were in high school, we heard a lot about it in the media. So it's where we mostly hear of anthrax as a biohazardous weapon. But anthrax is also a natural microorganism found in the soil, and it can infect many farm animals by getting into their hooves, getting into cuts within their feet. And this anthrax toxin essentially can kill them. So let's look at the anthrax toxin itself. So we see the anthrax toxin, the way it associates with the cell. We have an anthrax receptor, and we work into the cell with the PA unit, this protective antigen, and we assemble this unit together that allows for the LF and EF factors. That's the EF is the edema, the LF is the lethal factor, to attach. Once we have attachment of LF and EF on the outside of the cell, it essentially gets engulfed or endocytosed. When we endocytose these toxins, essentially the anthrax toxins, they're going to be able to be into the cell and then released into the cytoplasm of the cell. So what these actually do, the EF that we see here, the edema factor, causes fluid loss. So we can see that the cell is going to lose fluid into the extracellular space, so cells can die from this. And then we see a lethal factor as well that can destroy the regulatory cascade of the immune system. So it prevents the immune system from alerting that there is a problem, that there is an invader. So the edema factor can continue as the lethal factor prevents the immune system from catching this reaction. So that's how anthrax toxins work. Let's also go ahead and look at an endotoxin. So the endotoxin we're going to be looking at is called LPS. It's associated with gram-negative microorganisms. So it's only with gram-negative microorganisms. Remember, they have two membranes and a small peptidoglycan layer. And LPS is our lipopolysaccharide, and we actually see it right here. This is the LPS. When we're looking at LPS, it's not hazardous when it's on the cell itself. It's hazardous when it's released from the cell. So if you take an antibiotic that kills gram-negative bacteria, we take a specific kind of antibiotic that kills them in a certain way, it can release LPS into your system, and it can cause essentially this a toxic shock from LPS. So it can give you fever, it can cause death and result in the endotoxins to be released into your blood, as we see here. So LPS in low concentrations is not necessarily a problem, especially when it's in living cells, but it's only when it's in a large quantity of cells and then released from the membrane upon the death of the gram-negative cells. So it's something we have to think about for shock factors when LPS is released into the blood, that it can cause shock for humans. Okay, I think that's about everything.